This protein is derived from a crystal structure of bovine rhodopsin, and it's taken from the organism Bosporus, and not Homo sapien, but the structure of the two proteins are very similar. So we can see here the protein structure folded, the secondary structure folded into tertiary. So in red, we can see the alpha helices, and there are 7, which are the 7 transmembrane domains. In green, these are connected by random coils, and then in yellow, you can see some small beta pleated sheets as well. We can also see the B factor, which gives us some information about the motility of different parts of the protein. So in blue, it says low motility, and this is in the membrane. So that's expected because there's not much movement. But at the bottom, at the C terminus, we can see that there's a lot of movement shown in red. And that suggests that this part of the molecule is what interacts with different molecules to allow for rhodopsin to carry out its function. So the G protein coupled receptor is responsible for visual transduction is called meta-rhodopsin 2. And activation of meta-rhodopsin 2 opens the groove on surface receptor to bind the transducin alpha molecule. And this causes signal amplification and then ultimately visual transduction. Now, this process is terminated when the subsequent receptor phosphorylation mediates displacement of the G protein alpha subunit by arrestin and terminates signaling and therefore inhibiting visual transduction. And these are the references.